Now let's try a more complex query and count how many variants in our table are rare variants and are also located within a disease-associated gene. To do this, we need to identify the appropriate column names. To classify rarity, we will use the minor allele frequency of the variant for European Americans in the ESP project. And for disease association, we will access the disease phenotype information that was derived from the ClinVar database. Let's start with the query. The number returned to us is 7. We can now change the query such that the results that are returned to us are no longer a number, but the actual gene name. We can use the up arrow to use the previous command and simply change the count operator to the appropriate field name, which is gene. You should see all 7 genes printed to screen. There are many complex queries that you can do based on the different annotations that are provided within Gemini. We encourage you to take a look at the Gemini manual and explore the database schema. The above examples illustrate ad hoc queries that do not account or filter upon the genotypes of individual sample. Let's move on and try querying using the genotype information that is available in Gemini. Genotype information, for example, genotype, the depth, and the genotype quality, for each variant is stored in Gemini using a slightly different format, and so the syntax for accessing it is also altered. To retrieve the alleles for a given sample, one would need to add GTS to the subject ID to the select statement. For all other information, the prefixes, which is followed by the subject ID, are as follows. For example, GT underscore types, GT underscore depths, or GT underscore quals. Try the following query and add in the -- header flag so that we can keep track of what each column is referring to. We will also pipe to head so that only the first few lines get written to screen. For the rare variants, let's get the genotype for subject number 4805. We'll also obtain the depth and the quality of the aligned sequence so that we can assess the confidence in the genotype. If we wanted, we could also display information for all samples rather than typing out each subject ID, and we could do that using the wildcard character. Here we're just looking at sample number 4805. Often, we want to focus only on variants where a given sample has a specific genotype. For example, we're looking for homozygous variants within a family of trios. In Gemini, we cannot directly do this in the query, but the Gemini query tool has an option that's called --GT filter that allows us to specify filters to apply to the returned rows. Let's try an example where we report genotypes of variants within subject 4805 that have high quality genotypes in all samples, where quality is defined with an aligned depth greater than or equal to 50. When you're using the filter command, you need to take note of the specific syntax that is required. This is all listed in the Gemini manual, and we encourage you to take a read. So you see here, we now have the genes that meet our criteria printed to screen. We have the genotype for sample 4805, and we also have the reference and alternate alleles listed. What we could have done is also listed the actual quality scores to ensure that we did in fact filter by a quality score of greater than or equal to 50. Gemini also has a number of built-in tools, which incorporates the pedigree structures that was provided in the form of a PED file. Using this information, we can go a step further and query within a single family to identify disease-causing or disease-associated genetic variants reliably from the broader background of variants. In our example, we will make use of the autosomal dominant tool to query the trio of samples that we have been working with thus far. This tool is useful for identifying variants that meet an autosomal dominant inheritance pattern. The reported variants will be restricted to those variants having the potential to impact the function of affecting protein coding transcripts. Our PED file indicates that within our trio, both the mother and son are affected. And since we have only one of the parents to be affected, the tool will report variants where both the affected child and the affected parent are heterozygous. We'll query and limit the attributes returned by using the -- columns option. Let's clear the screen before we begin this query. The first few columns that are returned include family information, genotype, and phenotype for each individual. But all other columns are the same fields that we have been using in our previous examples: the chromosome number, reference and alternate alleles, the gene name, and the impact. We can filter results using the -- filter option, which would serve as the where clause as if we were using Gemini query. For example, we could search for only variants of high impact. And here we have listed only high impact variants. In order to eliminate less confident genotypes, we can always add the -D option to enforce a minimum sequence depth for each sample. Additionally, if we had multiple families in our dataset, we could specify to Gemini the minimum number of families for the variant to be present in using the --min kindreds option. Alternatively, we could select the specific families that we wanted to query using the --families command. With time, the query language will become more familiar to you, allowing you to really harness the power of Gemini to obtain a clearer functional interpretation of your variant calling results.